Hello everyone. I am Dr. Shruti Sethi and I am working as scientist in the Division of Post-Harvest Technology in Indian Agricultural Research Institute. In continuation of the teleconference on the course on food microbiology, I will be taking up the block 2, that is control of microorganisms. I will be dealing with the various contents like principles of preservation against microbial spoilage of food, the classification of foods based on pH, and the control of microorganisms by thermal processing, freezing, drying, and chemicals. Now, why do you want to control microorganisms? Like Mrs. Vijay Sethi had told you that the microorganisms are both our friends as well as foes. We can produce different products with the help of microorganisms which will add variety to our diet, but also on the other hand they are leading to spoilage of our food products. So therefore we require to control the undesirable changes caused by the microorganisms. As she had told you that there are various principles of preservation against for preserving the food products, and they are principle of preservation against the decomposition caused by microorganisms, then principle of preservation against decomposition caused by chemical changes, and thirdly preservation against the attack by rodents and insects. I will be dealing with the principles of preservation to control the microbial spoilage of food. This you can attain by 4 methods. That is, firstly, to keep out the microorganisms. This can be done by the use of aseptic conditions or aseptic processing. Then you can also have preservation against microorganisms by the removal of microorganisms. This you can do by the process of sorting out. That is, you remove any infected portion of the food product or the spoiled outer covering of the food product so that it remains stable during storage. Then you can also remove the microorganisms by the process of washing. You can either wash with running water or you can wash it with chlorinated water, that is around 3 to 5 ppm of chlorine. Then also you can use the method of filtration to remove the microorganisms. Nowadays microbial filters are there which will help you to remove the microorganisms from the fluid products. The third process for the principle for the preservation against microbial spoilage is hindering the growth and activity of microorganisms. This you can do by the help of low temperature, drying, anaerobic conditions, or by the use of chemicals. Then also preservation can be attained by the killing of microorganisms. This you can do by either thermal processing, that is sterilization, pasteurization, or the use of radiations. Generally what is done is you are using a combination of all these methods. It is called as combined effect preservation technology, or it is also called as hurdle technology. So the basic approaches for food preservation are that you have different processes, that is removal of microorganisms by method of filtration, centrifugation, then use of low temperatures, that is refrigeration or freezing temperatures. This will help to reduce the activity of the microorganisms. Then you can have high temperature application, that is pasteurization, canning, sterilization. Also you can have the approach of reduced water availability, that is you are reducing the water available for the microbial growth. So you can, for this you can use either different solutes like salts, sugars, or you can go for the process of drying, that is freeze drying, drum drying. Then you can also go for chemical preservation. Herein you are using generally recognized as safe chemicals for the preservation of food products against microbial spoilage. Then you can use radiation processing. Either you can use ionizing radiations such as gamma radiations, which will help to penetrate into the food and kill the microbes, or you can also have use of non-ionizing radiations such as UV rays. These are used in food processing industries so as to sterilize the packaging material as they don't have any penetration effect. Then also you can have the use of microbial based preservatives, that is use of bacteriocins, acids, etc. Now coming to the classification of foods based on pH. On this basis you can classify the foods into 4 categories, that is low acid foods which have a pH level of above 5.3, and these include corn, lima beans and other vegetables. Then you have medium acid foods which have pH range between 4.5 and 5.3. Then you have acid foods having pH between 3.7 and 4.5, and high acid foods, that is pickles and sauerkraut, which is having pH more than 3.7 or less than 3.7. So on basis of this pH, you have different processing parameters to attain shelf stability of the food material. And you can see in this diagram that 4.6 is the demarcation pH. Below this pH, that is low acid, high acid foods and acidic foods, you require mild heat treatment. That is, you can process the foods at atmospheric temperatures. The pH is having synergistic effect along with the heat, so it helps to reduce the spore growth, or it prevents the spore growth. And the foods which is having pH above 4.6, that is low acid or medium acid foods, they have to be processed at under pressure. That is, you have to do pressure processing, like in case of canning, because these foods, they are having low acid, so it is not having a synergistic effect. So you have to do pressure processing for these food products. Now here in this table you can see that the microorganisms, that is bacteria, yeast and mold, they are having different pH for activity. That is, they have a minimum, optimum and a maximum pH. And bacteria have a range of 4.5 to 9. Molds have 1.5 to 11 and yeast have 1.5 to 8.5, with the optimum being 6.5 to 7.5 for bacteria, that is near the neutral values of pH, and molds and yeast can grow at a slightly acidic conditions also. Now coming to the different methods of preservation to control the microbial growth, I will take one by one. First we will deal with the thermal preservation of food products. Thermal preservation is nothing but the application of heat to destroy the microorganisms. So there are degrees of thermal processing which can be applied to the food so as to destroy the microbes or to inactivate or destroy the enzymes which are causing spoilage in the food product. So first is the process of sterilization. Sterilization is the heating of the food products at a temperature of 121 degrees centigrade for a period of 15 to 20 minutes, and this will lead to the complete destruction of microorganisms. So you can have completely sterile food products which are shelf stable, that is, you can keep them for a long time. Then you have a process of commercial sterilization. This is usually referred in the case of canned food products, and this under this condition all pathogenic and toxin forming organisms are dead. That is, commercial sterility is obtained, that is both vegetative cells and spores, they will be destroyed and they will not be able to grow until and unless favorable conditions are provided to these products. The next degree of thermal processing is pasteurization. Pasteurization is generally done at a temperature below 100 degrees centigrade, and it destroys all the pathogenic microorganisms. Pathogenic means that they are disease causing microorganisms. So, but the pasteurization process, it will not lead to the extension of shelf life for a longer time. You have to refrigerate the pasteurized product always. You cannot keep it outside. So refrigeration has to be done along with the pasteurization process. And then there are different degrees of pasteurization, that is high temperature long time, low temperature long time, high temperature short time and ultra high temperature heating. In case of low temperature long time heating, that is LTLT process, you are heating the food at 63 degrees centigrade for a period of 30 minutes. And in case of high temperature short time, it is used for products which have an effect of thermal process on them, that is either nutritive value may be degraded or off flavor or off color is developed, like in case of citrus juices, or in case of milk. HTST process is preferred and it is done at 72 degrees centigrade for a period of 15 seconds. Then there is ultra high temperature. This is also carried out for milk, in which burning may be avoided. So it is done at a temperature of around 135 to 140 degrees centigrade for 1 second. The next degree of heat processing is blanching. It is usually done before the freezing process or the dehydration process, and its main purpose is to inactivates the enzymes which cause browning during storage, and this browning is caused by polyphenol oxidase enzyme, which may lead to the unacceptable color of the food product to the consumers. So blanching is used, and it is usually carried out at a temperature of 100 degrees centigrade with the use of boiling water, or you can also use steam in this case, and it will help to reduce the microbial load, plus it will inactivate the enzyme, and it will keep the fruits and vegetables in proper condition. Then cooking is also done in households. That is also a method of preservation, but it is also for a shorter period of time. The product will remain stable only under refrigerated conditions. Now in this graph you can see that how the microorganism growth pattern is there. You will see that the microbes, they grow in an exponential pattern. Like you will have microbe growing in with various proceeding of generation. They will grow in numbers like 1, 2, 4, 8, 16 and 32. And when you plot a curve of the log number of microbial cells or with the time, you will see that it will give a linear curve. That is, the microbes are growing in a linear fashion, in an exponential manner. And in the arithmetic growth curve, you can see it is a J-shaped curve, and it is the number of cells are growing along with the increase in time. Now coming to the rate of microbial death, when you are applying any thermal process, the microorganisms, they usually die at a constant rate, as you can see in the graph which is being shown to you. The graph is plotted with the log number of microorganisms at the y-axis and time in the x-axis. So as you keep on giving heat treatment, with the increase in time, the microbial growth will decrease at a constant rate. And the curve is plotted, that is thermal death time curve. It is the curve plotted between the time in log versus temperature at the x-axis. It will help to determine the process time, that is the thermal process time to kill the microorganisms present in food. According to these thermal death curves, you can see that the heat kills the microorganism logarithmically. That is, if 90% are killed in the first minute at a certain temperature, then in the next minute, this 90% of the surviving microorganisms will be killed. And then again in the third minute, again the remaining microorganisms which are there, again 90% of them will be killed. So it is a constant process. And spores, they are found to be more heat resistant as compared to the vegetative cells. The spores, what are they? They are the dormant state of the microorganism. That is, they are formed when you are giving any unfavorable condition to the microorganism. So the spores which will be formed, they will be in dormant state, and when you give favorable conditions, they will be again growing to the vegetative cell. So when under unfavorable conditions, these spores will be formed and they will be resistant to heat, cold and other environmental changes. Whereas vegetative cells, they will die because of the unfavorable conditions. Now, according to the heat resistance of microorganisms, you will see that thermophiles are more resistant as compared to the mesophiles, which are even more resistant than psychrophiles. This is because thermophiles have a higher optimum temperature for growth and multiplication. So therefore, if you are applying heat thermal process, thermophiles will be more resistant as compared to the psychrophiles. Then spore formers, they are found to be more resistant as compared to non-spore forming microorganisms. And the cocci, they are more resistant as compared to rods. And it has been found that the microorganisms that clump together, or they form cell aggregates, or those which have capsules around them, they are more heat resistant as compared to those which do not have capsules, or those which do not grow in clumps. Then microbial cells which are growing in a high lipid media, they will be more resistant to thermal shock as compared to those which are growing in low lipid media. This is because the lipid content is having a protective effect on the microorganisms. Then we have various terms associated with the heat preservation. Like you have thermal death point. It is the lowest temperature to kill the bacteria in a broth in 10 minutes. Broth is a liquid suspension. So it is the lowest temperature required to kill all the bacteria in 10 minutes. And thermal death time is a time required to kill all the bacteria in a broth at a given temperature. And you can determine the TDT, that is thermal death time, by the decimal reduction time, or D value. Here I have shown you that what is D value. So when you plot the log number of survivors at the y-axis with time at the x-axis, you will get the survivor curve. Which is a straight line. So this survivor curve, when it traverses one log cycle, the time at which it traverses one log cycle, that is known as the D value, or that is the 90% reduction in the microbial population. Then the other graph you can see is the Z value. Z value is obtained from the graph, that is the thermal resistance curve, which is plotted between the log D values and the temperature. So the temperature span which is required for reduction in one log D value, that is termed as the Z value. Here I am giving you the definition of D value, Z value and F value. That is, D value is the time required to reduce the population of microbes by 90%, that is 10-fold reduction, at a specified temperature and under specified conditions. Z value is the change in temperature in degree centigrade or degree Fahrenheit necessary to cause a 10-fold change in the D value of an organism under specified conditions. And F value is also in the time in minutes, but at a specific temperature of 121 degrees centigrade, which is needed to kill a population of cells or spores. Now we have a 12D process. It is used as a margin of safety in case of canned food products, which are usually of low acid, that is, which have a pH of more than 4.6. This is because you have to prevent the growth of microorganisms such as Bacillus coagulans. So this is the time temperature process that will reduce the Clostridium botulinum spore population by 12 log cycles. Like D value is by 1 log cycle, you are applying 12 Ds, that is reduction by 12 log cycles. And in this process, enough heat is provided to reduce 10⁻12 spores of Clostridium botulinum to 1 spore per ml. Now here in this graph you can see this pictorial representation that if you have initial high microbial load, it will take longer time to destroy all the microorganisms in this case. But if you have a lower initial microbial load, it will take the thermal process will take a lesser time to kill all the microorganisms. So